So good afternoon to everyone. It's a great pleasure to host the 7th webinar of the Young Scientist Program's webinar series of the Societies of Biophysics, Biochemistry, and IUPAB. So today we have the honor to have 4 women speakers, and one of them is my colleague of department, Professor Kowaltowski, and I introduce the other young scientists when they are about to speak. So to mention a few words about Professor Kowaltowski, she got a medical doctor and a PhD from Unicamp and went for a postdoc at Oregon Graduate Institute. And she became a very young faculty at my department. And she has been having a wonderful career as academic, as a professor. She's now a member of the Brazilian Academy of Science. She has written 2 books, one in English and another in Portuguese. And I don't want to say about how many papers, citations, and prize she has won, because this is too much and would take a lot of time of the science she is eager to talk to us. So without further delay, I give you the speech, Professor Kowaltowski. I'll just stop sharing.
Thank you, Mauricio, and I'll share my screen. Let me know if there's any problem seeing my slides here. So first of all, thank you for inviting me to speak here. I have to thank IUPAB-SBBF and my home, SBBQ, for speaking today. And particularly, I have to thank you for allowing me to speak in a young scientist forum, because I think through these very, very difficult times, what has really maintained our sanity as professors, at least for me, are the young scientists in our lab, are the wonderful and talented people that we have the honor to work with. So I feel very fortunate to be speaking here, and then followed by 3 wonderful and highly selected young scientists. So my lab has, for many years, been working on mitochondrial effects in different age-related diseases. And I want to tell you a story today about how mitochondrial function, dietary interventions that interfere with aging, and calcium form a triad which is regulating metabolism and regulating cell fate. And all of this comes from an interest that our lab has for a number of years now, which is to study mechanisms in obesity. So we know that obesity is a very serious health problem around the world. It's a growing concern, and it's a concern that leads to lower lifespans, and more importantly, to lower health spans. There's an increase in age-related diseases when aging is associated with obesity. Now, we know this for decades already, and most people, if you ask them, would not prefer to be obese. Yet obesity continues. And the reason for that is that it's not very easy to prevent obesity just by sheer will. We are evolutionarily programmed to like food, to like high-calorie food. We can't erase millions of years of evolution that have now, in recent years, made us sicker and decreased our lifespans. So I don't think that just public health prevention is going to solve the problem of obesity. I think we've proven that already. I think we have to understand mechanisms behind obesity, and understanding mechanisms behind obesity, we can uncover new targets so that we can treat diseases related to obesity and health problems related to obesity in a much better manner. And that's where my lab works, and mostly working in mitochondrial bioenergetic mechanisms behind obesity, because that's what I am. I'm a specialist in mitochondrial bioenergetics. So we study obesity in many models, but one of the models that's very important to us, and that I always like to talk about when I give these talks, is the normal laboratory rodent. And the reason I like to talk about this is that I don't think most people notice when they use normal laboratory rats and mice that they're actually using an obese animal model. What happens is that when we feed our laboratory rodents, we typically feed our lab rats or mice ad libitum diets, which means they can eat everything they want. Now, typically they have pretty healthy diets in terms of laboratory rodent chow, which is quite low in fats. But these diets were designed to make these animals reproduce best and not necessarily to make these animals survive most. And the animals that eat these diets ad libitum actually exhibit most of the characteristics of a mildly obese human. So they'll have quite a bit of body fat weight. They'll have high glucose levels in their blood. They'll have high insulin levels. They'll have markers of obesity, such as inflammatory markers, typical of a human couch potato who is slightly overweight. And you can prevent all these markers of obesity in laboratory rodents by either restricting their diet, such as calorie restriction diets, which are what we do a lot in the lab, or by exercising these animals, which I'm not going to talk about today. So by restricting diets, and specifically by restricting calories in the diet without decreasing micronutrients, you can decrease obesity in these animals and make these animals look more like a healthy, normal human. Not only that, you can increase lifespans in these animals, and you can decrease age-related diseases in a manner very similar to the prevention of age-related diseases that you see in healthy weight humans versus obese humans. So by comparing ad libitum fed animals, which are mildly obese, to calorically restricted animals, which are lean, we can look for mechanisms behind obesity and look for mechanisms related to age-related diseases that are linked to obesity. So I'm going to tell you the story about what happens with calorie restriction in terms of 3 different cell types, 3 different tissues, just because they're related to each other. And I'm going to start with what happens with calorie restriction in beta cells and insulin secreting cells. Now, I think we all know that preventing obesity prevents type 2 diabetes. So it obviously affects insulin signaling and also affects insulin secretion from beta cells. However, very few studies have been done on what calorie restriction in an animal does to beta cells. And the reason for that is that beta cells are a very, very small part of an animal. There are very few beta cells per animal. They're hard to study because they're in the pancreas that tends to degrade these beta cells if you try to isolate them. And calorie restriction is a very difficult diet to implement. It's a chronic diet to implement. So I really have to take my hat off for my students here, because my students who do calorie restriction studies in these rodent models have to go to the lab every single day, weigh out 40% less of the diet of the control animal, an especially prepared diet that has extra micronutrients, so these animals don't have malnutrition, and then offer this diet daily to these animals for 3 or 4 months. And daily is every day, including weekends, holidays, pandemics. It's something you just can't stop because it's a very chronic treatment. So calorie restriction is a lot of work, and it's hard to study its effects in beta cells because beta cells are such a small part of the animal. In that sense, we have an advantage over most people in the area, which is that we adapted a protocol from Rafael de Cabo and NIA's group a few years ago, which allows us to study calorie restriction in vitro. What we do is we get rats and we treat them for 3 months with calorie restricted diets, and I'm going to call that CR from now on, or ad libitum diets, which I'm going to call AL from now on. So now we have either lean animals, the calorie restricted, or we have obese animals, which are the ad libitum animals. And after 3 months, we get these rats and we sacrifice them, get their serum, and use these sera instead of the serum that you normally use in cell culture. So if you culture your cell with FBS or whatever other serum that you normally use, we're going to substitute this sera for the sera of the calorie restricted or the ad libitum animal and look at what happens in these cells. And what we find, and many other groups find, is that many of the cellular effects of calorie restriction can be reproduced by these soluble factors that exist in the sera of these animals. So what we did here was look at either INS1 cells, which is a cell line that secretes insulin, or in primary beta cells, both in mouse islets or isolated beta cells from rats. I'm only going to show you the INS1 data, but the rest of the data is in the paper. So if you want to look at the data in the primary models, it's already published. And what we did was try to see what happens in terms of insulin secretion with these sera. and also try to look at mitochondrial physiology, because mitochondrial function controls insulin secretion. So the first thing that we did was treat the cells for 24 hours with sera from either the ad libitum animals, which once again are animals which are obese, and these are going to be the white columns, or the calorie-restricted animals, which are going to be the black columns, which are animals that have normal, healthy body. After 24 hours, we measured insulin secretion from these cells under two conditions. Under conditions of low glucose, 2 millimolar glucose, and under conditions of high glucose. Now, healthy beta cells are expected to secrete low insulin under low glucose and high insulin under high glucose conditions. And that's exactly what we see with the calorie-restricted cells, with the cells that were treated with a sera from the calorie-restricted animals. On the other hand, the cells that were treated with a sera from the ad libitum animals secrete more insulin in low glucose and less insulin in high glucose. This is quite surprising to us because just 24 hours with the sera of the animals that are either obese or lean changes insulin secretion in these cells. So there are factors in the sera that lead to changes in insulin secretion, and these changes are acute, which I think is really interesting. Now, why do these changes happen? If you remember your undergrad classes, insulin secretion is very dependent on mitochondrial function. Basically, what happens in the beta cell is that ATP production by oxidative phosphorylation is going to depend on the concentration of glucose in the blood. Higher glucose concentrations result in more glucose 6-phosphate and more ATP production by mitochondria. Lower glucose results in less ATP production by mitochondria. And these levels of ATP are going to control the activity of potassium channels and calcium channels that are going to lead to the secretion of insulin. So the more ATP production you have by mitochondria, the more secretion of insulin. So looking at mitochondrial function is interesting in terms of understanding what's happening in terms of insulin secretion. And we look at mitochondrial function by following and measuring mitochondrial oxygen consumption. So these are oxygen consumption rate measurements. The higher the numbers, the higher the rate of oxygen consumption. And this is a quantitative way to measure mitochondrial function within intact cells. We do this with an extracellular flux analyzer known as the Seahorse analyzer, which is a good method to measure oxygen consumption rates in small amounts of plated cells. What we see is that the quantity of oxygen that's consumed by the cells under low glucose conditions is very similar if these cells are incubated in sera from the obese ad libitum animals or in sera from the lean calorie-restricted animals. However, when we add high glucose, we can see that there's more oxygen consumption in the cells that were incubated with a sera from the calorie-restricted animals, and you can see a quantification here. This indicates that there's more oxygen consumption related to ATP production in these cells. These cells actually produce more ATP with high glucose, which is compatible with those higher levels of insulin release that we see. And we know that this oxygen consumption is related to ATP production because if we inhibit ATP production with oligomycin, we no longer have any difference between the two cell types. So cells incubated with sera from lean animals are capable of producing more ATP and having more oxygen consumption related to ATP production in the presence of high glucose. These cells that were treated with a sera from the lean animals also have higher maximal respiratory rates. So we can maximize respiration with an uncoupler such as FCCP. And when we measure the maximum electron transport rates in these cells also have higher maximal electron transport rates. So they have better electron transport, better ATP production, and that's why they secrete more insulin, these cells that were treated with the sera from the lean. Now, why does that happen? Simple answer would be if they have more mitochondria. But we actually measured markers of mitochondrial mass in these cells, and we cannot convincingly find changes in the mass of mitochondria per cell. Another reason for more ATP production, more efficient ATP production, could be because of changes in mitochondrial dynamics. So in the last few years, it's become very evident, even though this has been known for more than a century, that mitochondria are very dynamic organelles. Mitochondria are constantly changing size and shape, They're constantly moving around the cell, and they're also constantly undergoing fission, in which one mitochondrion becomes two mitochondria, and also fusion, in which two mitochondria become one mitochondria. And these processes of changing size and shape, changing locations within the cell, and exchanging components through fission and fusion are very important for mitochondrial function. There are actually correlations in the literature that show that lower mitochondrial dynamics are associated with lower efficiency of oxidative phosphorylation. So we asked if there were changes in mitochondrial dynamics that could explain that increase in ATP production and electron transport capacity that we see in the cells that were treated with Acera from the lean animals. To measure mitochondrial dynamics and exchange of mitochondrial components, what we did here, and this is a collaboration with Orion Shirahai, who developed this technique in his group, was we expressed a photoactivatable GFP in mitochondria. And this GFP is not green until you zap these mitochondria with a laser and you activate this GFP. And now you have green mitochondria, but you have green mitochondria only on one side of the cell, which was the cell that was photoactivated with this laser. What you can do now is follow the dilution of this GFP in the cell over time. And you can see that this GFP starts to distribute around the cell over time. And the dilution of this GFP is a measure of how much mitochondrial components are being exchanged across the mitochondrial network. What we found very consistently was that the dilution of this GFP protein, and therefore other mitochondrial components also, was 3 times faster in the calorie-restricted Sera-treated cells than in the cells treated with Acera from the obese animals. So we do indeed have much faster mitochondrial dynamics in cells that were treated with Sera from calorie-restricted animals. So what we're proposing in this work is that Sera from calorie-restricted animals, and therefore calorie restriction in general, leads to more mitochondrial dynamics, and more mitochondrial dynamics leads to more mitochondrial ATP production function. This leads to more insulin release because of more ATP within the cell. Now, if all that is true, we should be able to inhibit this whole process if we inhibit mitochondrial dynamics. So what we did was do a knockdown of mitofusin 2, which is a protein necessary for 2 mitochondria to fuse and therefore necessary to promote mitochondrial dynamics. And we went to look back again at what this does in terms of insulin release. So you'll remember from before that the relationship between insulin secretion with high glucose versus low glucose was much better, was much healthier in the cells treated with calorie-restricted Sera versus the cells treated with ad libitum Sera that have a poorer relationship in terms of increase in insulin secretion. When we look at this in cells that had mitofusin 2 silence, this difference completely disappears, which tells us that this increase in insulin secretion is actually related to this increase in mitochondrial dynamics that we see promoted by the Sera of the calorie-restricted animals. So overall, what we see is that increased mitochondrial dynamics leads to more insulin secretion, and more insulin secretion is associated with better insulin signaling in general in these calorie-restricted animals. Well, that's what I wanted to tell you about beta cells. I'm going to skip over now to the brain. The brain is probably the organ we most want to preserve during healthy aging. And the brain is affected by a number of diseases that are both age-related and obesity-related. These diseases include Parkinson's disease. These diseases include, very importantly, Alzheimer's disease, very strongly related to obesity. And these diseases also include stroke. All 3 of these diseases also involve a form of neuronal death and loss, which is excitotoxicity. So we have 3 neurological diseases that involve the same mechanism, and also all 3 of them protected by preventing obesity. So we decided to ask if calorie restriction could actually prevent excitotoxicity, and that was the reason why it prevented neurological damage in these 3 very different diseases. So what is excitotoxicity first, for those of you who don't work in neuroscience? So excitotoxicity is basically a toxic condition for neurons because they get overexcited. In a physiological condition, you will have the release of neurotransmitters activating the cell, leading to an increase in calcium. That's a physiological increase in calcium, and basically is what makes us think. However, in an excitotoxic condition, usually because there's lack of energy, there's a release of too much neurotransmitters. And if you have this over-release of neurotransmitters, you have an excitotoxic situation in which you have too much calcium entering the neuron, and too much calcium entering the neuron is toxic to this neuron and kills this neuron. And we hypothesized that we could have prevention of excitotoxicity by calorie restriction, just because so many diseases that involve excitotoxicity have a protection against them when animals or humans are lean. So to answer the question if calorie restriction could prevent excitotoxicity, we actually promoted excitotoxicity very directly by activating the entry of calcium into neurons using Kainic acid and comparing the neurotoxic effects of Kainic acid in animals that were either fed ad libitum or fed calorie restricted diets, in mice that were either fed ad libitum or calorie restricted diets. And we can follow the neurological effects of Kainic acid by looking at seizures in these animals. This is the outcome of this neurological damage. And the effect was enormous. We can actually see here that the time spent in a seizure state by the calorie restricted animals is much, much lower than the time spent in the seizure state by the ad libitum fed animals. So there's a lot of protection. And not only do they spend less time having seizures, the calorie restricted animals, when they do have seizures, they have milder seizures, which affect much less of their body. So if you look here, you can see that the seizures that the calorie restricted animals had were typically related to only one limb, while the seizures that the ad libitum, the obese animals had, could affect their whole body. So they also had more severe seizure states in addition to spending more time in the seizure state. So the first question I think is very, very much answered here. Calorie restriction strongly prevents excitotoxic cell death, and excitotoxic cell death promoted through a very direct mechanism of stimulating these neurons, overstimulating these neurons with Kainic acid. The question now is how calorie restriction prevents neurologic excitotoxic cell death. And here, of course, we thought of mitochondria. First of all, we thought of mitochondria because we always think they're the center of the universe in my lab, but also because mitochondria are well known to be involved in excitotoxic cell death. Mitochondria are involved in excitotoxic cell death because mitochondria can help buffer calcium within the cell. So calcium within the cell is typically controlled by plasma membrane pumps and by pumps in the ER. Pumps in the ER can remove calcium that came in through the plasma membrane, but the amount of calcium that can be accumulated in the ER is quite low. On the other hand, mitochondria can also take up calcium. They have less affinity for calcium than the ER, but they have a capacity for calcium take-up which is much, much larger than the ER. So mitochondria can act as sinks for calcium within the cell. And they do this by taking up calcium through a mitochondrial calcium uniporter, which is indicated here as MCU. And they can take up calcium to quantities that can actually precipitate within the mitochondrial matrix in the form of calcium phosphate salts, for example. Now, mitochondrial calcium accumulation is not infinite in every mitochondria. At a certain point, with an overload of mitochondrial calcium, and usually with oxidative stress associated to that, there's an opening of a non-selective pore called mitochondrial permeability transition pore. And this overload of calcium is now going to pour out of mitochondria through this pore, leading to an increase in calcium in the cell and cell death. So mitochondria can act as calcium sinks, but they're not infinite calcium sinks. There's a point in which they become too saturated with calcium, open these pores and throw the calcium out. And at the point in which mitochondria can no longer buffer calcium in neurons, that is the point in which excitotoxic cell death happens. So to make a very long story short, we decided to ask if mitochondria from calorically restricted brains were different in terms of mitochondrial calcium uptake versus mitochondria from ad libitum brains. So we got animals under these two diets and isolated brain mitochondria from them and measured calcium uptake by these mitochondria. We measured calcium uptake by mitochondria by incubating these isolated mitochondria in the presence of a calcium-sensitive probe that is not membrane permeable. So basically, we're measuring calcium levels outside, calcium concentrations outside mitochondria. Then we add boluses, and each one of these peaks is a bolus of calcium to this mitochondrial suspension. And we watch as mitochondria take up this calcium and remove the signal of mitochondrial, of non-extra mitochondrial calcium fluorescence from the system. And mitochondria are going to take up more and more and more calcium until they kind of become tired and the rate of calcium uptake starts to decrease. And finally, they stop taking up calcium. So from these kinds of traces, we can measure two different things. One is the calcium retention capacity, which is the ability to take up calcium, the total amount of calcium these mitochondria can take up per milligram protein mitochondria. And second is the calcium uptake rate, or the speed in which these mitochondria remove calcium after the first addition of calcium, so the initial calcium uptake rate. What we found was that calorie restriction very strongly increased by about 50% the total amount of calcium that mitochondria could take up, and also almost doubled the rate in which these mitochondria could take up calcium. So this in itself already answers because why our animals are so well protected against excitotoxicity. This demonstrates that the mitochondria of the neurons of these animals are capable of buffering calcium better in larger quantities and also a lot faster, therefore preventing the increase in calcium in the cytosol, which makes excitotoxicity happen. Now, why does this happen? This happens because mitochondria from calorie-restricted animals are less susceptible to the mitochondrial permeability transition, that pore I said that opens when there's too much calcium in mitochondria and makes calcium pour out. And we know this because when we treat these mitochondria with cyclosporin A, which is an inhibitor of this pore, these samples now behave exactly the same. So we know that by inhibiting permeability transition, ad libitum and calorie-restricted mitochondria behave in the same manner. And that means that basically the mitochondria from the obese animals are more susceptible to mitochondrial permeability transition. Now, I'm not going to go through all of this because, again, it's in the paper and this is published, you can read it. But we actually identified some modifications and signaling pathways that are involved in this prevention of mitochondrial permeability transition promoted by calorie restriction. This involves SIRT3 and acetylation of cyclophilin D. And by identifying these different components of this protection machinery against excitotoxicity, we've actually identified new targets that can be used to prevent damage by excitotoxicity in age-related diseases that are related to obesity. So this is something that makes me quite happy because we can now put this in the literature and other people can start thinking of these new pathways as pathways to try to prevent the effects of these horrible diseases. Another thing that really made me happy here, and I'm a basic scientist at heart, even though I did my undergrad degree in medical sciences, was that I believe we were the first to find that a dietary intervention changed mitochondrial calcium uptake. And this seems important to me because mitochondria are the center of most of energy metabolism. Calcium is a very important regulator of energy metabolism. We learn about it in our undergrad years. And diet is another very important regulator of energy metabolism. So it seems important to me that mitochondrial calcium is responsive to a dietary intervention such as calorie restriction. And this is something that we decided to focus on a lot in our lab after we got these results. And our first focus was to ask, well, does this happen only in brain mitochondria or does this happen in other mitochondria? And we jumped to a very different tissue from the brain, which is the liver, because the liver is very central in metabolism. And also there's a lot of regulation of metabolism in the liver by calcium. So we were interested to see if there was a change in calcium uptake with calorie restriction in liver mitochondria also. Did the same thing we did with the brain samples. We isolated mitochondria from livers of animals in calorie restricted diets, here now in gray, or ad libitum diets in black, and measured mitochondrial calcium uptake. And once again, we saw that calcium uptake was higher in the calorie restricted mitochondria versus the ad libitum mitochondria. So liver mitochondria behave similarly to brain mitochondria and are also changed in terms of calcium homeostasis by a dietary intervention. Is this important for livers? Mitochondrial calcium uptake is known to be determinant in the prevention of damage promoted to the liver by ischemia-reperfusion. So damage promoted by a liver attack, a lack of oxygenation in the liver. So we decided to measure what happens in terms of damage to the liver when we promote ischemia-reperfusion in ad libitum animals versus calorie-restricted livers. This here is the area that was damaged in the livers of ad libitum animals. So you can see that it's all whitish, and we painted this white line around it so we can show you the damaged area. This here is the damaged area in the calorie-restricted livers with the same intervention, with the same ischemia. We have an enormous prevention of ischemia in livers from calorie-restricted animals because we have this prevention of the damage promoted by excessive calcium in the mitochondria from the calorie-restricted animals. So once again, we have tissue protection associated with more calcium uptake in mitochondria from calorie-restricted animals. From these data, we could have assumed, because these were the two first tissues that we studied, that every single tissue leads to changes in mitochondrial calcium uptake and an increase in mitochondrial calcium uptake by calorie-restricted animals, in calorie-restricted animals. But it's not true, actually. When we decided to look at other mitochondria, such as skeletal muscle or cardiac mitochondria, we actually found that calorie restriction did not change calcium uptake. When we decided to look at kidney mitochondria, and this is ongoing work, we actually find that calorie restriction decreases calcium uptake in mitochondria, so the opposite effect. And finally, when we decided to look at different dietary interventions, such as fasting, for example, we found also that fasting overnight decreases calcium uptake into mitochondria. So overall, what that tells us is that mitochondrial calcium uptake is sensitive to dietary interventions. It's sensitive to different dietary interventions. And there are changes in mitochondrial calcium uptake that are dependent on the tissue in which these mitochondria are. But still, it seems that mitochondrial calcium uptake is an important and new finding in terms of the regulation of metabolism by a dietary intervention that prevents obesity. So what I told you until now is that in beta cells, we find that calorie restriction increases mitochondrial fusion and dynamics, and that this improves insulin secretion. I also told you that in brain, calorie restriction increases calcium uptake by mitochondria, and this protects against excitotoxicity. In liver, we also see a protection of the livers associated with less damage by ischemia-reperfusion, and this is related to an increase in mitochondrial calcium uptake. So we have changes in mitochondrial morphology and dynamics and changes in mitochondrial calcium, both being promoted by calorie restriction. Here we decided to ask, is this related? Is mitochondrial morphology somehow related to mitochondrial calcium? And is this really a triad? So here we did a very basic science question, which was, if we change mitochondrial morphology, do we change mitochondrial calcium? And to do that, we constructed cells with different shapes of mitochondria. So these are C2C12 cells under control conditions. They have pretty normal bean-shaped mitochondria like you see in Leninger's biochemistry. These are C2C12 cells with a dominant negative construction of DRP1, which is a protein that promotes mitochondrial fission. So if you inhibit DRP1, you get elongated mitochondria, kind of spaghetti-shaped mitochondria. And these are C2C12 cells in which we knock down mitofusin 2, which is necessary to fuse mitochondria, and therefore we get very small and ball-shaped mitochondria. We quantified this and proved that we really are changing the size and shape of the mitochondria, but we are not changing the area occupied by mitochondria per cell. So we still have the same amount of mitochondrial mass per cell, just different shapes. And under these conditions, we measured mitochondrial calcium uptake exactly the same way as we measured it before. And what we found was very consistently that mitochondrial size and shape does matter for mitochondrial calcium uptake. So average-shaped mitochondria take up calcium at average quantities and at average rates. Very elongated mitochondria take up more calcium at higher rates. And very small mitochondria take up less calcium at rather slow rates, so they have low uptake rates. So mitochondrial size and shape is determinant for mitochondrial calcium. What's the result of this for the cell? So a few years ago, Israel Seckler's group demonstrated that mitochondrial calcium uptake is important for a process in which cells repopulate their calcium concentration, a process called store-operated calcium entry, which is basically the entrance of calcium from outside the cell, replenishing calcium after cells have done a lot of calcium signaling. So we measured store-operated calcium entry in these cells with different shaped mitochondria to see if not only mitochondria were important, but to see if mitochondrial size and shape was important to repopulate calcium in the cell. And what we saw very consistently was that store-operated calcium entry was regulated by mitochondrial size and shape. So average shaped mitochondria will repopulate their calcium at an average speed. Larger, more fused mitochondria will repopulate calcium at a faster speed. You can see the quantification here. While very small mitochondria will repopulate calcium at a slower speed and will arrive at a lower final calcium level. These cells actually have lower calcium also in the ER and actually have ER stress. So what this tells us is that there's actually a triad in which calorie restriction and dietary modulations change mitochondrial morphology, and this is determinant for mitochondrial calcium. But mitochondrial calcium also changes cell calcium, which changes mitochondrial morphology. That's known from the literature. We know that increased fission in mitochondria decreases calcium uptake in these cells. More fused mitochondria take up more calcium, and this is going to regulate calcium in the cell and in the ER. Now, I am very excited about this data because I think there are lots of central things in biology, central phenomena in biology that have been shown to be regulated both by mitochondrial size and shape and by calcium in the cell. They have been shown these two processes separately. And when we find that mitochondrial size and shape is determinant for calcium in the cell, I think we may find that there are relationships between these two phenomena. For example, stem cell differentiation is determined by mitochondrial morphology and dynamics, also by calcium. Are these two related? I don't know. This is a question we still have to answer. Another process in which both these phenomena are found to play a part. Calcium is involved in fatty acid oxidation, and also mitochondrial size and shape and proximity to lipid droplets is involved in fatty acid oxidation. Are these two related? I don't know, but we have data in our lab that show that mitochondrial size and shape definitely plays a role in metabolism in cells, although not necessarily oxidative phosphorylation. So Paulo Kakimoto recently was very interested in studying what happens to the liver in high-fat diets and in lipid overload models in cell cultures. And it's pretty well known that when you treat liver cells with high levels of fatty acids, such as palmitate, you have mitochondrial fragmentation. You can see that very clearly here versus the more fused mitochondria in the control cells. However, and very surprisingly, what Paulo finds is that these very fragmented mitochondria generate ATP through oxidative phosphorylation just fine. There's no change in ATP production through oxidative phosphorylation. What she finds is that there is a very large change in the production of ATP by the glycolytic pathway, and that this is accompanied also by an increase in lactate secretion. Now, this is very weird. Why would a fatty acid increase glycolytic pathway and lactate secretion? Paulo finds that the regulation, and this is in a paper that just came out this year, I really stimulate you to read it because there's a lot of data there. Paulo finds that this is related to changes in redox state and that the glycolytic pathway is actually redox regulated. But I think that we can also hypothesize that possibly these changes in glycolysis could be related to changes in calcium levels, because calcium is a very central regulator of metabolism, including in glycolytic rates. So that's something that we still have to study in the lab. Another situation in which both calcium and mitochondrial dynamics are involved is insulin secretion, and we're studying this relationship in the lab right now. I'm not going to show you any data on that. And finally, in immunology, just about everything in the last few years became about mitochondria. So mitochondrial fission is involved and very important for M1 activation in many different immune cells. But calcium is also classically related to immune cell activation, and calcium signaling is involved in the activation of immune cells. And in this sense, Osvaldo Pereira, in his undergrad work, was interested in studying the activation of microglia, which are the macrophages of the brain, sort of. Microglia can be activated by stimuli such as LPS, which are going to imitate a situation in which you have an infection. And under these conditions, literature data had already shown that there's extensive mitochondrial fragmentation, which we also saw. What the literature hadn't looked at is if this results in changes in mitochondrial calcium uptake. And what Osvaldo actually found is that there's a very significant decrease in calcium uptake in the mitochondria of these LPS-activated microglias. This is accompanied by changes in calcium in the cell. So these cells with fragmented mitochondria and low calcium uptake into mitochondria have less calcium in the ER. You can see this by releasing calcium with capsigargan, and have less store-operated calcium entry, exactly like we see in the C2, C12 cells. Interestingly, the changes in mitochondrial morphology are responsible for these changes in calcium in the cell. And we know this because if we promote, without activating these cells, mitochondrial fragmentation just by inhibiting fragmentation with mitofusin-2 knockdown, we have a decrease both in ER calcium and in store-operated calcium entry. On the other hand, if we activate these cells but prevent the mitochondria from becoming fragmented, we reestablish the calcium in the ER and reestablish store-operated calcium entry. So mitochondrial fragmentation is very important to activate immune cells in general. This is known from the literature, but we're seeing that it's important to regulate calcium in these cells. And calcium is important for immune cell activation. So I think all these effects are all linked. So the linkage between these two phenomena is something that's really interesting to us, and we hope that this is something that we're going to have new findings within the next few years, and we're going to work a lot on that. And that's what I wanted to tell you today. I have to end by thanking all of you with a rather old picture from our lab. Hopefully we're going to be all together in the same place very soon to take a new lab picture. But I really have to thank the wonderful people and young people in my lab who have kept my hope up over these very difficult times, and also the funding agencies that have allowed us to be here. So thank you. I guess I'm
Congratulations, Alicia. Thank you very much. Wonderful talk. I have learned a lot of new things. You put a lot of new stuff there. Very nice. So let's see here. I open the question to everyone. I see if there is anything in the chat.
You want me to read here? I think there's 3 questions.
If you can, yeah, please.
Mauricio, if you want, you can just ask the person to turn the mic on and then ask the question. You know, that will be, I think, nicer.
Please turn your mic on and ask any question. Alicia is eager to answer, I'm sure about that. Well, I will start. Is there a question?
Mauricio, the Santu Hazard, are you there? Can you just turn your mic on?
I think these might be the people outside this room, aren't they? Because there's a question and answer and there's the chat. I'm not sure. Yeah, anyways, I'll answer the question. The question is that liver contains glucose 6-phosphatase, but muscle doesn't have this enzyme. Why? It's different functions for different organs. So muscle is not involved in the maintenance of glycemic levels, and therefore glucose that's fixed within the muscle doesn't leave the muscle cell anymore, and it's used for ATP production within the muscle.
So you're reading this Gonzalo Ferreira made 3 questions, right? In the Q&A.
Yeah, I didn't read his yet. It's kind of long here. I don't know if he's here.
Is Gonzalo there? Yeah. Gonzalo, are you there? Could you, Cíntia, pode só habilitar ou ler o microfone do Gonzalo?
Já passei ele para a playlist, ele está liberado.
Gonzalo, you can just turn on your mic now in camera if you want, and then ask your question. Yeah, Gonzalo's right.
Okay. Hi.
Hi.
Very nice to meet you. And thanks a lot for organizing these meetings. Thanks, Alicia, for your talk. I had two questions, not three, but those were related mainly to the fact that since the 70s, I think, that it has been established that with with restriction diets in calories, the T3 levels, right, the thyroid hormone levels decreases a lot. So maybe in the serum that you're applying to these beta cells, you have low T3. Then, because the T3 hormone acts through nuclear receptors and changes the level of expression at that level. So what genes do you think, if this could be involved in the things you see, exposing the cells to low serum, calorie restriction serums, if the T3 could be an intermediate step, and what genes could be affected? That would be the first question.
So yeah, one of our questions exactly is what's in the sera that leads to these changes. I have a postdoc working sort of on that. It's not easy because this is a natural product with a whole bunch of components, and everybody has their favorite component which could be doing this. And it's probably not a single component. But thyroid hormones are definitely involved in the differences that are promoted by these sera, along with a whole bunch of other hormones that are changed. So I think there are lots of different targets that could be involved. Typically, there's not a lot of thyroid hormones regulating ATP synthesis in beta cells. So it wouldn't be my first hypothesis, but I wouldn't discard it in any way because we know there are differences in the levels of these hormones. So that's something to look at.
Mm-hmm. Thank you, Alicia. May I ask the second question, Mauricio?
Yes,
Alicia?
go ahead. Yeah, please.
Okay. So the second question is related to the excitotoxicity response, because it is known that these mechanisms are somehow related to an hyperactivation of glutamate receptors like the NMDA receptor. And the NMDA receptor is quite permeable to calcium. So you're overloading the cells with calcium during this exocytotic response. So usually this excess of calcium will enter through the calcium uniporter system. So my question is if the CYR3 molecule that you have shown acts or changes the activity of the calcium uniporter, if you have any evidence about that, or another intake of calcium proteins carriers in the mitochondria, like the leucine, EFHAN, CPER motif. That could be another.
So we do have some unpublished indications that there may be regulation of the MCU complex by dietary interventions, but I don't think that's what's giving us these different results in our brain mitochondria. And the reason for that is that we don't have a change in calcium uptake rates when we treat the isolated mitochondria with cyclosporin A. And under these conditions, we're not changing the complex. We already isolated mitochondria. They're equal. We're only changing permeability transition, but we're equaling the rate and the capacity of these mitochondria. So it doesn't seem to be because of that. But we are looking at components of the MCU in mitochondria from different tissues with calorie restriction, because I'm definitely interested in that.
Mm-hmm. Thank you, Alicia. Thanks
Thank
a
you,
lot.
Gonzalo. Thank you, Alicia. There is another question. Santu, would you like to ask a question? I don't know.
There's a question written here. Does sonication solubilize both nuclear and mitochondrial membranes? I guess it depends on the sonication. We don't isolate mitochondria by sonication. We use sonication when we want to isolate different membranes from mitochondria, but we don't isolate them in that way. We use mechanical tissue breakdown, or we use detergent with cells.
So I have a question. I may be naive, so I apologize for that. I wonder if the calorie, the amount of calorie is really the thing that matter, or is one of the macronutrients that are making a bigger effect. So is this known?
So there are quite a few studies on that. And basically what we're doing is overall calorie restriction, which is mostly carbohydrate restriction because most of the diets of these rodents are carbohydrates. Our specific diet has no changes in micronutrients. So our animals are supplemented with vitamins and minerals to the same level as the animals that are fed ad libitum. So that's not what's involved. Now, other groups have studied different kinds of restrictions, and there are some restrictions that are very good in terms of increasing longevity, specifically restrictions of amino acids and methionine restriction specifically. So that might be one of the central involved species, and not just carbohydrates, even though carbohydrates is the most part of our restriction. Yeah, but we're just looking at overall calorie restriction. We didn't compare all these different diets.
Thank you very much. Is there any other question? So if not, I want to
Mauricio,
thank again.
there's a question in the chat. The chat tab.
So Clarissa asking, do you have data about electron transporters?
You mean electron transport chain components in the mitochondria from the different tissues?
Would you like to ask a question?
Animals? Yeah, okay. I think she has no voice. She's just writing things down. So yeah, there are changes in components of electron transport chain. There are changes in supercomplexes of mitochondria. It depends on the mitochondria, it depends on the dietary intervention, it depends on the tissue you isolated them from. So it's very complicated. There are tissue-specific changes. We published a number of years ago some data on differences in respiratory capacity and these changes, if you want to have a look at that. But there are also data from many other groups in that sense.
Thank you. So thank you very much, Alicia, again.